Dear friends, today we are going to discuss the topic low energy responses and high readiness responses of photoreceptors. The following are the main objectives of the present study. Number 1, to present a brief introduction about the light and its role in photomorphogenesis. Number 2, to give a brief account of different photoreceptors responsible for different photomorphogenetic responses with respect to light quality. Number 3, to study in detail the phytochrome mediated responses with respect to their fluence responses, that is energy requirements. Number 4, to discuss high readiness responses mediated by phytochrome. And number 5, to understand the role of low fluence responses in seed germination. And last, to know the role of blue light photoreceptors response for UVA and blue light absorption in HIR action spectrum. Now, let us have a brief introduction about the topic. Sunlight is the ultimate source of energy for virtually all life on Earth. For plants, light supplies the energy used in photosynthesis and signals used in the photoregulation of growth and development, that is photomorphogenesis. In photosynthesis, an energy transduction process, a bulk of which is carried out in the chloroplasts of higher plants, light energy is converted to and stored as chemical energy of photosynthetic products. Photosynthesis is the first fundamental step in the transfer of energy from light to food chains of the biosphere. Photomorphogenesis is the result of an interaction between light and living matter in which light is used primarily as a signal and not as energy source. Signal perception and transduction are the basic process in photomorphogenesis. The survival of organisms and species depends upon the ability to respond and adapt to the changes in the environmental factors. Photosensory systems that can acquire and process information about the light conditions, direction, duration, intensity, spectral quality of the environment are found in animals, fungi, microorganisms, and plants. The presence of photosensory system confers to the organism the capability to react with appropriate responses to the changing environmental conditions. Leaf orientation and chloroplast distribution within leaf cells change during the daytime in response to variations in the intensity and angle of incidence of sunlight, thus providing the best conditions for energy interception by the photosynthetic apparatus. Seasonal changes in the light conditions of the environment are reflected in seasonal temperature and precipitation patterns. Birds, insects and plants can apparently measure changes in the duration of daily illumination, that is photoperiodism, and processes such as migration, metamorphosis and flowering are carried out under conditions favorable for survival. In higher plants, photoregulation has been demonstrated for all phases of life cycle, that is from seed germination to seed formation, and for response at all levels of organization, that is from molecular to the whole organism level. The photoregulation of plant growth and development is the result of interaction between light and appropriate plant components. The basic component of the light sensing system are specialized molecules, that is photoreceptors, grouped into two categories. Number 1, blue light activated photoreceptors, that is cryptochrome and phototropins. Number 2, red and far-red activated photoreceptors, that is phytochrome. Number 1, cryptochrome. Cryptochrome is the blue light photoreceptor of plants, insects and mammals and plays an important role in the generation of circadian rhythms. Cryptochromes absorb in the blue spectrum with absorption peaks at 380 nanometer and 450 nanometer. Number 2, phototropins. Another blue light photoreceptor that has a major role in phototropism and chloroplast movement in phototropin. Chloroplast movement in leaves is an adaptive feature that helps to control light absorption and prevent photo damage. Number 3 is phytochrome. This photoreceptor absorbs both in red and far-red spectrum of light. Red and far-red spectra of light are involved in phytochrome-mediated photomorphogenetic responses of the plants. These include flower induction, stem elongation, chloroplast development, seed germination, etc. Phytochrome pigment is found to be present in two photoreversible forms, that is red light absorbing at 660 nanometer PR form, and number 2 is far red light absorbing form which absorbs at 730 nanometer designated as PFR form. The PFR form absorbs far red light and is converted to PR and inactive form. After absorption of red light, PR form is converted to active PFR form, while as PFR form absorbs in the far red spectrum and is converted back to inactive form, that is PR. This is termed as photoreversibility. Now let's discuss low energy responses and high irradiance responses of photoreceptors. Light controls diverse processes of plant growth and development. Early physiological and photochemical studies indicated that light responses in photomorphogenesis are classified as low or high energy reactions according to their energy requirements. Later, low energy reaction is induced by short pulses of irradiation with relatively small doses of light were divided into low fluorescence responses designated as LFRs, earlier called as low irradiance responses, and very low fluorescence responses designated as VLFRs, previously called very low irradiance responses. The high energy reaction which required a radiation with relatively high energy for relatively long period of time was renamed as high irradiance response. Phytochrome responses can be distinguished by the amount of light required. That is, phytochrome responses can be distinguished by the amount of light required to induce them. The amount of light is referred to as the fluorescence, which is defined as a number of photons striking on a unit surface area. The most commonly used units for fluorescence are moles of quanta per square meter. In addition to fluorescence, some phytochrome responses are sensitive to irradiance or fluorescence rate of light. The units of irradiance in terms of photons are moles of quanta per square meter per second. Each phytochrome response has a characteristic range of light fluorescences over which the magnitude of the response is proportional to the fluorescence. These responses fall into three major categories based on amount of light required. Number 1 is very low fluorescence responses, number 2 low fluorescence responses, and number 3 is the high irradiance response. Now what are very low fluorescence responses? Some phytochrome responses can be initiated by fluorescences as low as 0.1 nanomole per meter square and they saturate at about 50 nanomoles per meter square. That is, in dark grown owned seedlings, the growth of coleoptile has been shown to be enhanced by red light, and the growth of the mesocotyle, that is the elongated axis between the coleoptile and the root, is inhibited at such low fluorescence responses. Another example is that of seed germination in Arabidopsis. Seed germination has been induced in Arabidopsis by red light in the range of 1 to 100 nanomoles per meter square. These remarkable effects of vanishingly low levels of illumination are called very low fluorescence responses. Typically, such low levels of light are comparable to 1/10 of amount of light emitted from a firefly in a single flash. That is, only less than 0.02% of the total phytochrome is converted into PFR form by VLFRs induced through small amount of light. Because the far red light that would normally reverse a red light effect converts 97% of PFR form to PR form, about 3% of the phytochrome remains as PFR. That is significantly more than is needed to induce very low fluorescence responses. Thus, far red light cannot reverse very low fluorescence responses. That is, very low fluorescence responses are not photoreversible in nature. The very low fluorescence response action spectrum matches the absorption spectrum of PR form, indicating that very low fluorescence response is mediated by phytochrome and supporting the view that PFR forum is an active form of these responses. Now, low fluorescence responses. The classical red-for-red photoreversible responses discovered by Hendricks and Bostwick et al. Are known as low fluorescence responses. These responses are mediated by phytochrome and stimulated by light dose in the range of 1 micromole per meter square to 1000 micromole per meter square. This is equivalent to about a 0.1 second exposure under dense plant canopy at lower end and about 1 second of full daylight at the upper end. These responses include most of the red-for-red photoreversible responses such as promotion of lettuce seed germination, promotion of deetiolation, inhibition of internode elongation, and photoperiodism, that is control of flowering, and loss the regulation of leaf movements. Low fluorescence response spectra for seed germination in Arabidopsis include a mean peak stimulation in the red region that is at 660 nanometer, and a major peak for inhibition in the far-red region is 72 nanometer. Both very low fluorescence responses and low fluorescence responses can be induced by brief pulses of light, provided that the total amount of light energy adds up to the required fluorescence. The total fluorescence is a function of two factors. Number one, the fluorescence rate, designated as moles per meter square per second, and the irradiation time. Thus, a brief pulse of red light will induce a response provided that the light is sufficiently bright, and conversely, very dim light will work if the irradiation time is long enough. This reciprocal relationship between fluorescence rate and time is known as law of reciprocity. Very low fluorescence responses and low fluorescence responses both obey the law of reciprocity. Now, the role of fluorescence responses, both very low fluorescence responses and low fluorescence responses in induction of seed germination. Light is important ecological factor that modulates germination in large number of species. Depending on the spectral composition and photon fluorescence, temperature, water availability, and the physiological conditions of the seeds, it can promote or inhibit seed germination. In many cases, the stimulus of germination shows a biphasic response to the fluorescence of red light. The first phase, called as the very low fluorescence response, is present in seeds with extreme sensitivity to light. Very low fluorescence response is mediated by phytochrome A and is usually saturated by PFR over P total ratio that is lower than 0.1%, which can also be established by a far-red light pulse. Thus, they do not show the classical red far-red reversibility. In natural conditions, establishment of very low fluorescence response may occur when the seeds remain for a period of time in the soil. Very low fluorescence responses can also be achieved in dry stored seeds by laboratory treatments like chilling treatment, heating, temperature shifts, or incubation in presence of GE3. By contrast with very low fluorescence responses, the second phase or low fluorescence response is saturated by moderate to high PFR to P total ratio, such as those established by red light and are reversed when red light is immediately followed by far red light. The main photoreceptor involved in low fluorescence response is phytochrome B. Most light requiring seeds have tissues, for example the endosperm, that offers considerable resistance to the expansion of embryo. In these seeds, the control of germination lies in the balance between the capacity of embryo to grow and the constant imposed by the tissue that surround it. In Datura ferox, the stimulation of germination by low fluorescence response involves the weakening of micropylar region of the endosperm, designated as Me. Endosperm softening mediated by low fluorescence response is associated with an increment in Menon degrading activities in Me of tomato and Datura ferox. In Datura ferox, red action on the me involves both promotion of endo-beta-manase and beta-manosidase activities and the accumulation of endo-beta-manase and expansionis transcriptis. PFR formation also promotes embryo growth potential in lettuce, and in Datura ferox, this is associated with seed germination. Now, what are high irradiance responses? Phytochrome responses of the third type are termed as high irradiance responses, several of which are: number 1, responses such as leaf expansion and stem elongation under natural environmental conditions characterized by relatively high energy fluorescence rates of sunlight over a long period of time. Number 2, synthesis of anthocyanin in various dicot seedlings and in the apple skin segments. Number 3, inhibition of hypocotyl elongation in mustard, lettuce and petunia seedlings. Number 4, induction of flowering in henbane hyoscyamus. Number 5, pillular hook opening in lettuce. Number 5, enlargement of cotyledonous in mustard. And number 6, production of ethylene in sorghum. High irradiance responses require prolonged or continuous exposure to light of relatively high irradiance, and the response is proportional to the irradiance with a certain range. High irradiance responses are proportional to the irradiance as well as the duration. The reason that these responses are called high irradiance responses rather than high fluorescence response is that they are proportional to irradiance. Loosely speaking, the brightness of light rather than to fluorescence. High irradiance responses saturate at much higher fluorescence than low fluorescence responses, at least 100 times higher, and are not photoreversible, because neither continuous exposure to dim light nor transient exposure to bright light can induce high irradiance responses. High irradiance responses do not obey the law of reciprocity. Thus, high irradiance responses generally have the following characteristics. Number 1, full expression of the response required prolonged exposure to high irradiance, primarily with high promotion of far red light. The magnitude of the response is a function of the fluorescence rate and duration. Like very low fluorescence responses, high irradiance responses are not far red photoreversible. High irradiance responses do not obey the law of reciprocity. The photoreversible low fluorescence responses, particularly those involved in de-etiolation, also qualify as high irradiance responses. For example, at low fluorescences, the action spectrum for anthocyanin production in seedlings of white mustard shows a single peak in the red region of the spectrum. The effect is reversible with far red light, and the response obeys the law of reciprocity. However, if the dark green seedlings are instead exposed to high irradiance light for several hours, the action spectrum now includes peaks in the far red and blue regions of the spectrum. The effect is no longer photoreversible in nature, and the response becomes proportional to the irradiance. Thus, the same effect can be either an low fluorescence response or an high irradiance response, depending on its history of exposure to light. The involvement of phytochrome in the photocontrol of the final expression of many high irradiance responses has been demonstrated by the red far red reversibility, by the action of brief exposure to red light applied either before or after exposure to continuous irradiation. In several cases, a small but measurable amount of response whose full expression requires prolonged irradiation can be induced by a single brief exposure to red light. The effect of red light can be reversed by far red. In cabbage seedlings, the synthesis of amount of anthocyanin as high as 40 to 45% of that formed under continuous 24-hour far red irradiation can be induced by 4 brief 3-minute exposures to red light applied at 6-hour intervals. The action of red light can be reversed by brief exposure to far red applied immediately after each red light exposure. In some cases, the effect of brief red irradiation and that of prolonged exposure to far red are quantitatively very similar. For example, the effect of night interruption on flowering of short day plants, the induction of germination in light requiring seeds of the Bromeliaceae, and the elongation of arena coleoptiles. The germination of dark germinating varieties of seeds of lettuce, tomato and cucumber can be inhibited by prolonged exposure to far red light. The extent of the inhibition is a function of the irradiance and the duration of exposure. The effect of prolonged far red light treatment can be reversed by a terminal brief exposure to red light unless the exposure to far red light has been too long. The flux of red applied as a single brief exposure at the end of the far red treatment required to repromote germination are a function of irradiance and duration of exposure to far red light. The higher the irradiance and or the longer the duration of far red exposure, the higher the fluxes of red light required to promote germination. This effect of continuous far red light on the inhibition of germination because of its dependence upon irradiance and the duration of exposure can be considered as a true high irradiance response. Intermittent far red, for example 1 minute far red every 10 to 30 minutes is as effective as continuous far red inhibiting seed germination. Germination is also fully inhibited under intermittent red far red treatments. For example, 1 minute red followed by 1 minute far red every 10 to 30 minutes. Full germination is obtained under intermittent far red treatments. For example, 1 minute far red followed by 1 minute red every 10 to 30 minutes. The equivalence between intermittent and continuous treatments and the full red far red reversibility shown in the intermittent treatments indicate that phytochrome is probably the only photoreceptor involved in the photo control of this particular HIR response. There are some similarities in the action of intermittent light treatments on the inhibition of germination and on the synthesis of anthocyanin in young seedlings. Intermittent red far red treatments are more effective than intermittent far red in inhibiting seed germination. Intermittent red far red treatments are more effective than intermittent far red for the synthesis of anthocyanin in cabbage, mustard and turnip seedlings. These results demonstrate that the involvement of phytochrome in the photo control of the final expression of many high irradiance responses. But perhaps with the exception of seed germination results and those cases in which a brief red light exposure and a prolonged far red light exposure one produces responses quantitatively very similar. These data do not prove definitively that phytochrome is only pigment involved in the photo control of high irradiance responses. The high irradiance response action spectrum of etiolated seedlings has peaks in the far red, blue and UVA regions. For studying high irradiance responses such as inhibition of stem or hypocotyle growth, dark grown seedlings, etiolated seedlings have usually been selected, and the study has revealed that the main peak of activity for high irradiance responses is in the far red region of the spectrum between the absorption maxima of PR form and PFO form. Besides, these peaks also fall in blue and UVA regions of the spectrum. At first, researchers believed that another pigment other than phytochrome may be involved, as the absence of peak in the red region is unusual for phytochrome mediated responses. A number of evidences support the view that phytochrome is the one of the photoreceptors involved in high irradiance responses. On the other hand, a separate photoreceptor with peaks in the UVA region and the blue regions of the spectrum has been assumed to be responsible for the absorption in the UVA and blue regions of the spectrum. For the confirmation of these results, the HIR action spectrum for the inhibition of hypocotyle elongation was determined in dark grown HY2 mutants of Arabidopsis having little or no phytochrome holoprotein. As expected, the wild type of seedlings exhibited peaks in the UVA, blue and far red region of the spectrum. In contrast, the HY2 mutant failed to respond to either far red or red light. Although the phytochrome deficient HY2 mutant exhibited no peaks in the far red region and it showed a normal response to UVA and blue light. These results demonstrate that phytochrome is not involved in the high redness responses to either UVA or blue light, and that a separate blue/UVA photoreceptor is responsible for responses to these wavelengths. More recent studies indicate that the blue light photoreceptors, that is cryptochrome 1 and cryptochrome 2, are involved in blue light inhibition of hypocotyl elongation. With this, we will conclude the topic. Thanks for watching.